All right, after the half an hour has gone by, what you will see is the solution of SDS plus cells will actually be quite clear. So you can see how that solution is really clear, and it's because the cells have lysed. There are no clumps in there, as opposed to, here's another tube I made up for you of an unlysed solution. Right, so you can see the difference. This one's clear, the cells are lysed. This one's cloudy, the cells are not lysed. We want the clear lysed one because the DNA will be in this solution. Next, we take the solution and we're going to apply it to a spin column. So a little bit about spin columns. Spin columns are little tiny columns used to collect, concentrate, and clean up DNA. So they'll come pre-sterilized. So this one's packaged up in this little thing. I can pop it out. And there is a collection tube. Right, there's just an empty tube, no lid. And then here is the spin column. And the spin column does have a lid that opens and closes. So the white part is a silica gel packing material in the column. And silica gel is really good at binding negatively charged DNA. And that's good because it's going to help us concentrate all the DNA from the solution, the cell lysate. And then it'll also grab up proteins and lipids and other sort of random debris and junk. But we're going to be able to wash the spin column once the DNA is attached there. So I would label my sample. So this is E. coli. I'll just put EC on there for the sample. And I'm going to put the spin column back in its collection tube. And I'll open it. And then I'm going to put the whole thing into a rack, right, to keep everything upright. I'm going to take my blue pipette and my DNA containing solution, my cell lysate, should be about 500 microliters in volume. So 500 or, actually 520, because there'll be a little bit of an increase in volume from the cells themselves. Load a tip. And then I go into my solution, my cell lysate, and I pipette it up. Go down to the first stop, put the tip in, draw up all that material. It might be a bit sticky or stringy from the DNA released into solution. I'm done with this tube, so I'll discard it. And I'm going to load this onto the spin column. And it goes, close the lid, eject the tip. Now I've got the solution sitting on top of the spin column. It's not actually like into the filter yet, filter or column. And I want to push the DNA solution through. So the way that I do that is by centrifugal force. You can't balance, or you can't run a centrifuge if the tubes are unbalanced across the rotor. So I've got another tube here that weighs the same as the first tube. And I checked that before I started making this video. So I made sure they were roughly the same weight. And I'm going to use this just as a balancing tube for this. So both of these are going to be loaded into the centrifuge. Let me show you that. Into the centrifuge. So I've got my two samples in the centrifuge loaded across from another in a perfectly straight line. I put the lid on the centrifuge, and then I'm going to spin this sample at 10,000 G or 10,000 units of centrifugal force for 1 minute. So I preset the centrifuge. I can start it, and it makes a bit of a racket, but you can see the settings here. That will spin for 1 minute, and that will push the fluid through, but all the DNA will bind to the silica gel in the spin filter.